This is Dr. Dave Seftel reporting from AAO 2015. We're here today with Professor Sachs. First of all, Professor, tell us a little bit about the work that you're doing at your university and how it relates to retina implant.
So when we started the project in the mid-90s, it was a lot of animal research we did in the lab. And in 2005, we started the current project with patients. So this was done in different places, and most of the patient work is done at the University of Tübingen, and we did our first implantations there in Tübingen as well. And so now we are in the phase where we do a multi-center study, and that's where different sites, like the site where I'm working now in Dresden, are involved.
So tell us where you are in terms of an update to where we were last year at the same time.
So concerning the last year's work, we now try to get the lifetime of the implant longer. So this is the development of the implant itself, so that we reach a lifetime which is about 5 to 10 times longer at that implant that we were currently using in the project. And so we did our first implantations with this new implant with pretty good results, and we hope that we could meet these expectations now.
When you say pretty good results, what are we talking about in terms of the usable duration before you get rejection or you run out of power or various other technical impediments?
So it's not rejection or things like this. It's more a technical problem that you have a very sophisticated electronic in a very hostile environment in the subretinal space, and that it creates the lifetime problem for the implant. And so we were limited to about 1 year with the first implants, and now we think with the new implant we getting to 5 to 10 years, but this is what has to be tested right now.
So what are you seeing in the subretinal space? Are you getting a lot of fibrous reaction around the implant? What is the nature of the hostile environment that you're referring to?
We don't get fibrosis, things like this. This is some kind of a pretty good space to implant foreign body. On the other hand, what I mean with hostile environment, if you put electronics into a salty liquid, you always create problems. And that's what we have here. So it's merely a problem of passivation, which is not a simple problem, and this seems to be solved right now so that we have an acceptable lifetime. And acceptable lifetime is due to the fact that this implant can be exchanged as well. So if we have a lifetime of 5 to 10 years, and with 1 or 2 explantations or implantations again, so I think we have a chance to create a situation for the patient which is really acceptable.
What are the implications for reimplantation? So the device reaches its natural, expected technical and biological lifespan, and then you have to put another one in.
Well, you see, in the first phase of the project, we saw these lifetime problems, and we are glad to have this exchange option. And concerning the problem of reimplantation, this is surgically or technically a thing which is I want to say very easy to do, but it can be done with a great safety level, so it's not a problem for us to do a reimplantation.
So in terms of European regulatory approval, where are you in the current path towards approval of this device?
So this implant which we had for the study, which we published in Vision Research right now, was an implant which was approved by the European Community, has the CE mark, and the new implant we were talking about with the longer lifetime is currently at the step to get this CE mark right now.
When do you expect that you will have enough data for the EMA to proceed?
You mean for the CE mark?
Well, for the clinical approval of implantation.
I think within the beginning of the new year we have the chance to have this, but this is always something which is unsure because it's administration problem.
Right. As you look at the life cycle of these devices, what is the current resolution that you're able to accomplish, and where would you like to be in terms of resolution in the future?
The resolution, I think, is merely a question of the distribution of the ganglion cells. So with the implant, we come very near this possible resolution. So there may be a factor of 1.5 or about that size that we could get higher, but we are very close to this size right now. So maybe there will not be implants which give us the chance to read tiny little text. This seems not to be possible at the moment.
Right. But that's still very impressive that you've been able to accomplish that level of resolution. And when you mention the ganglion cells, which ganglion cells are you actually stimulating? Are these in the visual cortex? Are they at a different side of the brain?
What we stimulate is the ganglion cell in the retina, mainly the bipolar cells. So if we come from the subretinal space, it's that what we expect to stimulate right there.
Okay. So obviously this innovation presumes an intact optic nerve and a functioning optic nerve.
Definitely yes. Without an optic nerve, without a functioning optic nerve, it's not possible to get this kind of stimulation.
Right. So for patients who've lost their optic nerve either due to infection, inflammation, or assault, there are no projections to essentially put the wiring directly into the visual cortex.
So that's quite true. So this is only for retinal degeneration, but this is a huge amount of patients which can be helped by this. The first target was the RP. It was a good model for testing this. Whether there might be other indications is something for the future.
Right. Professor, is there any additional things you'd like to share with us about retina implant, about the work, and about what the future holds?
I don't think there is a lot which I could say here. I want to emphasize that the subretinal implant is not a camera-based system, which is, to say it in simple words, a more physiological seeing, because the object that you your percept is definitely at the point where you expect it.
Right.
And with a camera-based system, it's not, and that makes the great difference.
Right. So you're relying on the pre-existing lens and optical mechanism, and you're focusing on the photoreceptor portion, replacing that with an active and working and functioning high-resolution photoreceptor.
That's right. We simply substitute the photoreceptor which has been gone, but it's nothing which is in the higher level. And this is the advantage of this project, because you definitely get a retinotopic stimulation, which is what we expect when we want.
This is a very, very exciting area that we have followed for some time, and we're thrilled that Professor Sachs is here to share the latest progress with us.